Hello and welcome everyone to this podcast. In this podcast, we're going to talk about the different stages of photosynthesis. In the last podcast, we introduced some of the terms for photosynthesis and the chloroplast, and I drew a picture of it. And so here is that picture, not the one I drew, but the one from your textbook. So I want to remind you what's happening here in the chloroplast. Photosynthesis can be broken up into these two stages, stage 1 and stage 2. In stage 1, we have light and water entering into the system. We'll talk in a moment how it will take electrons and transfer them along the way to produce ATP and NADPH, producing oxygen. Then, when we're done with that, we'll move on to stage 2 and how stage 2 uses ATP and NADPH in the carbon fixation cycle. You may have learned it at some point as the Calvin cycle, same cycle, and how that is used to generate sugars, amino acids, and fatty acids. So now, let's look at stage 1 and stage 2 and provide a little bit more information about these two stages. In some ways, stage 1 is similar to oxidative phosphorylation because they both use electron transport to take energy from the electrons to pump protons across a membrane. Now, as they do that, they will use that to make ATP using that enzyme we talked about last time called ATP synthase. Now, they are different in some ways. Oxidative phosphorylation is in the mitochondria, whereas photosynthesis occurs in the chloroplast, in the thylakoid. It will use the thylakoid membranes to pump those protons into the thylakoid space. Remember, mitochondria, they use the intermembrane and the intermembrane space. So this is a little different. In oxidative phosphorylation, we take electrons from the carriers of NADH. In photosynthesis, chlorophyll is used. And these chlorophylls absorb energy from photons from the sun. This is why we call this sometimes the light reaction. So remember, this is all generated from light. And that light comes from the sun. And I always find this amazing because the sun, right, is 93 million miles away. This giant star, so many miles away, is what generates stage 1 of the light reaction. Now, stage 2 is sometimes called the dark reaction and sometimes called the Calvin cycle, after one of the individuals who worked on and discovered this cycle. Okay, stage 2 uses the products of stage 1, ATP and NADPH, to convert carbon dioxide into sugar. I'll say it can occur in the absence of light, which is why we often will see it referred to as the dark reaction. The reason I don't particularly like calling it the dark reaction because it implies, or at least suggests, that this only occurs in the dark. And in fact, many of the enzymes that are found in the Calvin cycle, stage 2 here, are downregulated when there's no sun or no light. So let me write that down here. Many stage 2 reactions are off or inhibited at night, in the dark. So even though it can occur in the absence of light, often light is used as a feedback mechanism to make sure these enzymes are on or off. Okay, so let's spend a little bit of time talking about chlorophyll before we jump into a discussion of stage 1 photosynthesis. So chlorophyll is a pigment in the thylakoid membrane. This pigment absorbs light. Particularly, it absorbs red and blue light very well. but it absorbs green light poorly. So it's reflected off, and that's why most plants, when we see them, or photosynthetic organisms, when we see them, are green. Now chlorophyll, as you see in this picture here, has a unique structure. And this unique structure is what helps it absorb light and, more importantly, absorb energy that's going to be used in the various photosystems during photosynthesis stage 1. There are two main components of a chlorophyll molecule: the hydrocarbon tail, shown here, and the porphyrin-like ring structure. The hydrocarbon tail is important because it will help chlorophyll associate with other hydrophobic proteins. As you can see here, this tail is also very hydrophobic, and together it anchors with these other proteins. It anchors the chlorophyll molecule to the thylakoid membrane. Okay, let's talk about the porphyrin-like ring structure next. The porphyrin ring-like structure here contains around it what is often referred to as an electron cloud. Not really a cloud, but there's just a lot of electrons here that are easily excitable. So electrons that are easily excitable. So what happens is a photon from the sun will interact with this electron cloud and excite the electrons in it. So I'll write that here: photon excites electrons in chlorophyll. It makes those electrons present as a higher energy. These excited electrons here are unstable. So the excited electrons in chlorophyll are unstable. Okay, so now these excited electrons, being unstable, they want to be able to release that energy. So if this were outside the cell, those excited electrons could be released and it would generate heat or light. However, inside the cell, inside a photosynthetic organism cell, we're going to harness that light energy, the light energy from the photon from the sun, and convert that into chemical energy. And that's what we're going to talk about next. The ability to capture light energy and convert it to chemical energy requires what is known as a photosystem. So photosystems in general, they capture light energy and convert it into chemical energy. Okay, so here's our thylakoid membrane, and within the thylakoid membrane is where the photosystems are located. Now these photosystems contain two main kinds of complexes. The first are the antenna complexes. The antenna complexes contain lots of chlorophylls. All these chlorophylls in here take advantage of the photons that come in here to destabilize them. You'll have different chlorophylls at different energy levels within a antenna complex, and they can be adjacent to each other, as I'm showing here. By having them so close to each other, when they want to transfer energy to their neighbors, it's easier to do that. And so why is it transferring energy? Well, remember, we said we have our very good friend here, the sun. It's shooting down photons here, and when it hits a chlorophyll molecule, remember, it excites it to a higher energy. And remember, that higher energy makes it very unstable, and it wants to release some of that energy so it can become stable again. And it does so by passing it on to chlorophylls that have a lower energy. It can do it within a antenna complex, or it can do it between neighboring complexes. But the goal is to move that unstable energy, that high energy, from a chlorophyll molecule with higher energy, less stability, to one that has lower energy. And by moving it to one with lower energy, you reduce some of that overall instability. Which brings us to our next complex. And our next complex, I'm going to show here, is called the reaction center. Within the reaction center. So within the reaction center, there are these two special chlorophylls. And your book calls these chlorophyll special pair. So they're a unique pair of chlorophyll molecules. And what makes them unique is that of all the chlorophylls that we've seen so far, these are at the lowest energy. This is where that energy, this unstable energy from the antenna complex ultimately will move to. And at this point here, there are no other chlorophylls for it to transfer because we're at the lowest pair. So now what does it do with that energy here? It still wants to get rid of it. In fact, it has to get rid of it. So what's right next to this? So what's it going to do with that energy, those electrons? It's going to move those electrons to this special carrier. This is an electron carrier. As it does this movement, this is going to become negative. And up here, this special pair of chlorophylls will become positive. We'll come back to this in a little bit, because with this being positively charged, we have to regenerate it so that the next time electrons need to move to here, they can. But I'm not going to talk about that for a little bit, so you have to keep watching to figure out what actually happens there. The suspense must be killing you, I'm sure. Now before I go to a new board here, I want to keep this picture here because there's something I want to come back to in just a moment. So I want to mention something down here in the corner. So let me box this off here. And I want to remind you that photosynthetic organisms' goal, or one of their main goals, is to build organic molecules like sugars. To accomplish this, the plants are going to need lots of energy. And they're going to need lots of reducing power. They get this energy in the form of ATP that they're going to have to generate. And the reducing power, they get in the form of NADPH. In order to make both ATP and NADPH, they're going to require two photosystems. This part's going to be the same regardless. How the electrons are excited, how they're transferred to this special pair of chlorophyll molecules, and then how they're passed to this carrier. Now what's going to change between one photosystem to the next is the type of electron carrier here and what the end product will be. So we're going to talk about two photosystems next. The one that makes ATP, which is the first one, is called Photosystem II. And the one that comes after that, the one that makes NADPH, is called Photosystem I. Now it may not make sense that the first photosystem is called 2 and the second photosystem is called 1. It's for historical reasons. The first one here was discovered first, so it's going to be called Photosystem I, as well as the Photosystem II was discovered second. So it has nothing to do with the order, it's just kind of how they popped up in their discoveries. Alright, I'm going to erase this and we're going to start talking about Photosystem II. Okay, so let's talk about Photosystem II. Let's begin it again by drawing the thylakoid membrane. And we can draw our photosystem here with the antenna complex and then the reaction center next to it. So here I'm going to just write AC for antenna complex and then reaction center RC, just so we keep up with everything that's being written here. And the same thing is going to happen as we had on the last whiteboard. We have the sun sending its photons down, exciting these chlorophyll molecules in here, where it's going to transfer those electrons after they've been excited to chlorophylls of lower energy states until it finally reaches that reaction center where we have these special pairs of chlorophylls where the electrons are finally transferred to. And I said on the last slide that there was a protein that was adjacent to the reaction center that acted as an electron acceptor here. So they would go here and this would be negatively charged, and then these chlorophylls here would be positively charged. Now for Photosystem II here, this electron acceptor is called plastoquinone. I shouldn't draw an arrow here. It implies it's moving. It's not moving. I'm just labeling it plastoquinone. It then, just like we've seen before, will transfer these electrons to something that has a higher affinity for the electrons. And in this case, it'll be this protein complex called cytochrome B6F complex. Using this energy from the electrons moving through, it's able to move protons through this complex. So we end up then with this higher concentration of these protons on this side of the thylakoid. Still in the thylakoid. This is in the thylakoid space. And just like we saw with oxidative phosphorylation, we have this ATP synthase with the stalk here that spins around and releases ATP. And it releases ATP just as you would suspect by having these protons moving through here. Just like we saw with oxidative phosphorylation, that causes the ADP to be converted to ATP. So as I said on the last whiteboard, we needed to make both ATP and NADPH. So this is how we effectively make ATP. And we're going to use this ATP in just a moment when we talk about stage 2 of photosynthesis. Again, these electrons are excited. We use that energy of these high energy electrons to pump protons across this gradient, and then the protons then go with the gradient and move across the ATP synthase, generating ATP. So next we need to make NADPH, and to make NADPH we're going to have to look at Photosystem I. Alright, so Photosystem I. Again, I'm going to draw the thylakoid membrane. We're still going to have the photosystem here, where we have the antenna complex and the reaction center adjacent to it. We're still going to have the sun here. Can't get rid of the sun. It's still going to excite the electrons in just the same way. Those electrons found in these chlorophyll molecules. It's going to transfer these electrons just like we've talked about before, this high energy chlorophyll, to a lower energy state chlorophyll molecule until it winds up with the special pair of chlorophylls. And at this point here, as we've said before, these electrons need to get out of here, but this is already at the lowest state, so they're going to go and transfer to that electron carrier that I mentioned before. In Photosystem II, it was plastoquinone. In Photosystem I, it is a protein that we'll abbreviate calling it FD, but it's spelled out ferredoxin. Again, it'll take the electrons like so. In doing so, this becomes negatively charged. This special pair becomes positively charged. And as promised, I'll come back to that in a little bit. This will then take those electrons to this complex here called ferredoxin, NADP reductase. And it will convert NADP plus to NADPH. So photosystem 1 takes that energy from this high energy electron, passes it to ferredoxin. It then takes that that electron to ferredoxin NADP reductase, where it generates NADPH. And as we said before, the whole purpose of photosystem 1 is to make NADPH, which will then go on to stage 2, which we'll talk about in just a moment. Okay, now we want to talk about what I've promised you I'd come back to, the regeneration of the special chlorophyll pair. And we'll talk about Photosystem II first. And so I'm going to draw our obligatory thylakoid membrane. I'm not going to draw out the whole photosystem, but I do want to draw out the reaction center. And remember when we had these chlorophyll pairs here, and they transferred that electron to plastoquinone. And in doing so, I said this generates a positive charge on these chlorophyll pairs, and a negative charge here. Now we need to regenerate this special pair, so we have to add electrons to it. And the way it's going to do this is it's going to use an enzyme here that I'm going to call simply just a water splitting enzyme. And what it will do, as you might have guessed from the name of it, it's going to take two water molecules and it's going to generate oxygen. So remember, in our equation for photosynthesis, we're generating oxygen, and this is where we're generating the oxygen. And the reason the oxygen is being regenerated is just to recycle this special chlorophyll pair. As the water is being split and you're making the oxygen here, electrons are going to flow in here to the chlorophylls, removing that positive charge. Okay, so again, in regenerating this special chlorophyll pair, water is used to generate oxygen, and the electrons released from that are given to these chlorophylls. Okay, so this is how we regenerate the special chlorophyll pair for Photosystem II. Now, what about Photosystem I? You have that reaction center here, just like we saw with Photosystem II, where you had those special chlorophyll pairs here that have that positive charge that we need to get rid of. So it can be regenerated and used again for the next cycle of passing that electron down the road here. Now, interestingly, it's going to get the electrons to remove that positive charge from Photosystem II. So let me explain how that happens. So remember this plastoquinone here. It's going to pass those electrons to cytochrome B6F complex. And remember, it does that in the process of moving protons across the complex. And that's kind of where I ended the story when we talked about this before. And I just left this little electron just hanging out here. So what happens to it? Well, it's picked up by a protein called plastocyanin. So PC, I'll put here. Those electrons move to this plastocyanin, and then those electrons are pushed over here to this special chlorophyll pair of Photosystem I to remove that, or to neutralize that positive charge. So now they can be used again for the next round of electron transfer. Okay, so that ends our discussion for stage 1, the light reactions of photosynthesis. Remember, we made oxygen, which will be used by oxidative phosphorylation by the plant and other organisms on Earth. Important for stage 2 here, it generated ATP and NADPH. It's important to note that it's incredibly important, essential, that ATP and NADPH are made in the thylakoid membranes and not somewhere else. Because NADPH and ATP, they can't pass through this membrane. So that's good for a couple reasons. One, we're not going to lose it. It's going to be devoted to the carbon fixation cycle. But we also can't get it from the outside. And if we can't get it from the outside, it has to be made in here. So without stage 1, we wouldn't get ATP or NADPH to fuel the carbon fixation cycle. So let's talk now in greater detail about the carbon fixation cycle, how we're going to use ATP and NADPH with CO2 to generate sugars, amino acids, and fatty acids. Okay, so let's talk about carbon fixation now. And there's a whole cycle I want to mention, but I want to really focus in on this very first step here. And this first step is where the actual carbon fixation occurs. And it's going to take a 5-carbon molecule called ribulose 1,5-bisphosphate. And remember, I said it's a 5-carbon structure. It will take a molecule of carbon dioxide and this fascinating enzyme called rubisco. And I'm going to come back to rubisco in a moment. But rubisco is going to fix this carbon from the CO2 to this molecule. And it's going to make what we're just going to call a 6-carbon intermediate. This 6-carbon intermediate then will become 2 molecules of 3-phosphoglycerate. Each of these molecules is 3 carbons. So you start off with 5 carbons, you add a carbon from CO2 to become 6 carbons, This short-living intermediate is then converted into 2 3-carbon molecules, phosphoglycerate. Now this reaction, I should state, is largely unfavorable. The way that this reaction becomes more favorable is by having a constant supply of ribulose 1,5-bisphosphate. As long as this is being regenerated at a constant rate, the reaction will become favorable. So how are we going to replenish it? Well, we're going to replenish it by having this carbon fixation cycle, the Calvin cycle. Now, as we proceed with the cycle, I'm going to start off with 3 CO2s and 3 ribulose 1,5-bisphosphates. And because of that, I'm going to end up with, but with the 3 phosphoglycerates, I'm going to end up with 6 of those. So to get the cycle going, these 3 phosphoglycerate molecules, sugars, will be converted into 1,3-bisphosphoglycerate. In doing so, for these 6 molecules of 3-PGA here, we're going to consume 6 molecules of ATP, producing 6 molecules of ADP. These 6 molecules of 1,3-bisphosphoglycerate are then converted into 6 molecules of glyceraldehyde, 3-phosphate. In doing so, 6 NADPHs are consumed and turned into 6 NADP pluses. Now this is why I started off with 3 here so we could generate 6. Because for every 6 that are made, 1 is released from the cycle. So one of these glyceraldehydes, 3-phosphates, is released. And then it can be released from the chloroplast to make more complex sugars, fatty acids, and amino acids. The 5 remaining glyceraldehyde 3-phosphates, so now we have 5 of those, they are going to be used to regenerate ribulose 1,5-bisphosphate. In doing so, 3 ATP will be consumed, producing 3 ADP. So if you add up here, we have 15 carbons here because we have 5 3-carbon molecules here. And here we're going to end up with 3 5-carbon molecules. So it's just a reorganization of the bonds to regenerate this ribulose 1,5-bisphosphate. And as long as this is being regenerated, Rubisco can act upon it to fix the carbon and continue the cycle over and over again, and thereby making this whole reaction a favorable reaction. Okay, let's spend a little time talking about Rubisco. It's one of my top 3 favorite enzymes of all time, so that's saying something, I guess. It has some very interesting features. The first is, without Rubisco, we get no sugar, so it makes sugar. So without this, we never generate this glyceraldehyde 3-phosphate, so we never generate these complex sugars. We don't make the plants, the crops, the big trees that we see because we're not making these sugars. This tiny molecule carbon dioxide is responsible for making the tallest trees, the smallest shrubs, everything. And then animals rely upon these plants, all this complex sugars that these plants are made of, for their survival. But speaking of just the enzyme itself, it's got a couple features we should mention. It is an incredibly slow enzyme. I'm not sure where it ranks amongst all enzymes, but it is a very slow enzyme. The conversion of ribulose 1,5-bisphosphate to this intermediate to the 3-phosphoglycerate is very slow. But this being a very important step here, we can't have it be too slow. So the way it makes up for that is that Rubisco is roughly 50% of all proteins found in the chloroplast. There's a lot of proteins, a lot of enzymes in the chloroplasts, but roughly 50% of all of that is Rubisco. So it's slow, but it's mighty. There's a lot of it there. It is also, I guess I have to put it over here, the most abundant protein on Earth. So it's a slow one, but it's the most abundant. Okay, now I want to summarize what is made in carbon fixation and what is consumed. So carbon fixation summary. Okay, so for every 3 CO2s that enter the cycle, 1 molecule of glyceraldehyde, 3-phosphate, is made. Now, it makes 6 of these molecules, but the other 5 get recycled. This is an actual product because it leaves the cycle and is used to make those complex sugars, or simple sugars. In doing that, for every 3 CO2s, 9 ATP are consumed. 6 in the beginning of the pathway of the cycle, 3 at the end of the cycle. We also consume 6 NADPH are consumed. So it's a rather energetically demanding cycle to generate this, what might appear to be a fairly simple 3-carbon structure. But this is what gives ultimately that plant all the structure it needs to survive, and ultimately all the sugar secondary and tertiary consumers require as well. Okay, that's all I have for stages 1 and 2 of photosynthesis. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.